Sollito, and I'm a postdoc at the University of Florence at the Rockefeller University. Today, I will talk about the need to generate a streamlined pipeline annotation and the effort to implement this in the Galaxy platform. So, as part of the VGL lab and together with the Wellcome Sanger Institute, we are included in the Vertebrate Genome Project. That's a project connected with the Earth Biogenome Project, which is a global initiative aimed at sequencing, cataloging, and studying the genomes of known eukaryotic species. The aim of the VGP is to produce error-free reference genomes and the related gene model annotation for all the 70,000 existing vertebrate species. All the data produced and related to the generation of reference genome will be then included in an open access repository, which is GenomeArk. This serves as a central database for the reference genome of vertebrates and even of other eukaryotic species, offering to researchers, conservationists, to the public access to these genomic resources. And why there is the need to produce high-quality assembly. Of course, this is an important step for performing downstream analysis, such as for phylogenetic analysis, always more accurate, and for adaptation and evolutionary studies, for example, to study the evolution of chromosomes, and for comparative genomic analysis, and even for population and conservation genomic research. At the moment, we are at the end of the first phase of the project. In this phase, the requirement was to the generation of the assemblies for representative species from each of the major vertebrate orders. Up to now, one species for each order has been assembled with more than 500 species assembled, and 90% of them have been manually created, but only a small percentage, around 40%, have been annotated through the RefSeq annotation pipeline. That is the internal NCBI annotation pipeline. So the aim of this project is to link the genome assembly with the strong gene model annotation to scale up the process of annotation and in order to fill this gap. And however, predicting genes in large eukaryotic genomes is still a significant challenge. For that reason, we need the development of new algorithms and or new streamlined pipelines. To finalize this project, 3 different actors are involved: us as VGL, the Comparative Genomic Resources Group of NCBI, and the Galaxy developer team. To help to reach this aim, so to scale up the annotation process, Galaxy has a key and fundamental role. But I will just say a few words on what is Galaxy. Basically, it's a web-based scientific analysis platform which core values are accessibility, reproducibility, and transparency, which provides free computer resources for users who might not have computational power. It includes more than 8,000 tools at the moment, with the possibility to produce and generate reproducible workflows. A good example is the release of an article that has been published in 2024 with the latest VGP genome assembly workflow on Galaxy. It was published with the aim to produce and scale up the assembly process. So starting from different input data, for example, starting from iFi long reads, these go in a stage of profiling of the k-mers, and then these iFi reads will be assembled through iFiASM in a second stage of contiguing. There is a third step of purging the haplotypic bubbles, a fourth steps of scaffolding using IC or Bionano data if available, and the final step in order to reach the final assembly of high-quality assembly. With, of course, each stage has the specific evaluation processes. In this case, we were able to reach the final genome, and following the footprints of this workflow, we are now releasing the alpha version of the annotation workflow. So it's based on GAPX, that's the external NCBI annotation pipeline. which is based on an Xflow workflow management system. And on Galaxy, it requires two different ways to input the data. We can input the data through a precompiled configuration file in a YAML format, or directly putting the input through the Galaxy history. But which are the mandatory input data that we need to perform the annotation process? Basically, we need a masked genome that should be soft masked. So the genome should be soft masked by repetitive elements. Then we need RNA-seq data. The best should to have a high variability of expressed gene. So it's necessary to use starting different tissue ages and or developmental stages to ensure this higher variability in the genes transcribed. And of course, this would help to create a more complete and more accurate annotation of this genome. But of course, not always this kind of data are available. And the last mandatory input is the taxonomical identifier, which allows for searching the appropriate database of proteins. And if we go on checking the workflow, we see that starting from input data, that there are the one that we mentioned before. EGAPix use this data in order to produce the GFF file. This file, through the different tools of AGA toolkit, will be manipulated to make a standard version of the annotation file, to fix the overlapping genes, and to keep the longest isoform. In this way, we will obtain the final GFF file. This will go for some statistic and annotation metric, always through AGAT. So basically, we will have the number of genes, number of exons, the mean gene length, and the mean exon length, and other annotation metrics. And always through the AGA toolkit, we will be able to extract the protein sequences that will be evaluated through a Busco analysis with a specific lineage-dependent database in order to check the completeness of our annotation process. But if we go on the tail on the AGAPix process, it's based on the NCBI internal annotation workflow, that is RefSec. Some modules are already implemented of the RefSec. Some others should be released in the future version of the workflow. And as we can see, so the RNA-seq module, where we align the reads against the reference genome using a star, and then the alignments will be ranked, collapsed, and then filtered. This is possible, is already been implemented in the EGAPix workflow. The second module, is the module related to the protein alignment, where we align the proteins against the reference genome using the miniprot. And even in this case, the alignments are ranked and filtered. And then the third module that is already been implemented is where we, through the use of chainer that combines partial alignments into longer models, and through the use of Genome that processes the alignment data in order to generate the final gene models, we will be able to reach the accept.gff file, so the annotation file. Some other modules of the RefSec annotation pipeline still have to be implemented. As you can see, the transcript alignment module, the identification of non-coding RNA, small or long non-coding RNA through different tools, and the gene assignment. So basically, where we define each gene model with its specific gene name. So these models will be released in the future versions of the workflow in Galaxy. I test the workflow in Galaxy using a different reference genome, and here just report what has been done on Ctenoplegia guttata. That's an iconic species known even as zebrafinch. Its genome is at chromosome scale, as you can see from the N50, L50 values, so the contiguity levels values, and it presents 41 chromosome with a really high percentage of BUSCO completeness. On NCBI, there is already an annotation of phi that was annotated through the RefSec internal pipeline and performance started from around 500 RNA-seq dataset. These numbers are challenging for a lot of research groups, considering the massive data storage and powerful resources. But of course, having 500 RNA-seq dataset for not iconic or non-model species is really complicated. So we decide to reduce our dataset, and we use just 20 RNA-seq dataset coming from different tissues in order to maintain a higher diversity in the gene expressed. And as you can see, we use different tissue as brain, tongue, and so on and so forth. From the resources point of view, EGAPix required around 15 hours to end the complete process, with two different tasks that require a higher time amount than others. And these tasks are the alignment of the RNA reads with the star and the mini-prot alignment. But of course, these steps depends on the number of starting reads for star and the number of starting proteins that are present in the database for the mini-prot alignment. And if we check the physical memory, so the RAM usage of EGAPix, the same two tasks require higher RAM, but in this case, even the step where I build the index of the genome preceding the alignment with star requires an higher RAM usage, but of course, this is depending on the genome size. Checking the results, I performed a BUSCO analysis using the passeriforme dataset as a specific lineage on the proteins obtained from EGAPix and released from NCBI. I obtained quite comparable results. Indeed, in both cases, we have a high level of completeness, both considering the complete files or considering only the longest isoform for each gene model. And with the EGAPix, we have a higher number of missing genes rather than NCBI. So basically, EGAPix is a bit low performing. This, of course, can depend from the high amount of dataset used by NCBI and NCBI internal pipeline. So just to remind you, they use more than 500 RNA-seq dataset to perform the complete annotation. But if we check merely the numbers, we see that are still quite comparable, but considering Jacquard index, that's an index of similarity among two different datasets, we can say that at the gene level, the Jacquard index is quite high, it's 0.88, but at exon level is decreased. We have a value of 0.65. This means, and this can be dependent to the different splicing isoform, or so to exons that can be retained or skipped in different cases, but even to the exon shift. There is still need to improve and ameliorate this annotation workflow, and there is still need to still a room for improvements for that. And in conclusion, this workflow is enough for version. Of course, needs more tests and needs a lot of other implementation. Firstly, we would like to assign to each gene model a specific name, and we would like to identify the non-coding part of the genome. So where we identify the small non-coding RNA, the long non-coding RNA through different tools usage, such as TR-scan-C or, for example, Infernal. And we need to implement even the identification of pseudogenes. Related to the final part of the workflow, we would like to implement even the evaluation process. And however, I think we need to include even the possibility to use a custom protein database to perform the protein alignment that should be independent from taxonomical identifier. In fact, in this case, for example, for the zebrafinch is using the Sauropsida dataset, but maybe I'm using a more specific dataset, for example, if we go at the order level, so in this case at the passeriformes level, we can ameliorate our results. So please, if you want to test the alpha version, you can scan the QR code. Of course, you can run it through the Galaxy platform. And I would like to thank all of you, the Vertebrate Genome Lab, the University of Florence, of course, the Galaxy team, and the NCBI Comparative Genomics Group that helped me and support me in this research. And I would like to thank all of you. Thank you.
Hello, everyone. So my name is Mathieu Mufato, and I work at the Wellcome Sanger Institute. Today, I want to talk about a project called Genome Data Party. It's a public repository of standard genome analysis. So I work at the Wellcome Sanger Institute in the Tree of Life department. And you may know Tree of Life from sequencing a lot of genomes, but we're doing sequencing in order to increase our understanding of genomes and life in biodiversity. So those data are crucial, and the sort of analysis we can deliver on those data are crucial for to deliver the scientific questions that the different research teams are trying to answer. But really, this endeavor is shared with many other projects. So we are part of Darwin Tree of Life, which focuses on sequencing species in Britain and Ireland. But we're part of a few international projects as well, like the Aquatic Symbiosis Project, which is about marine species, Vertebrate Genome Project, and there's a hierarchy of projects. For instance, we are part of the ERGA and EBP as well. All of that, the ultimate goal is to sequence all life on Earth. So in the department, we are specifically in the delivery and operations team. We are multiple teams there, and I want to give a shout out to my team, first of all. So I got people working on data management creation. I've got some mathematicians and some people working in infrastructure, and all of us together are helping deliver those data, those projects of Sanger, and building this Genome Data Party repository that I'm going to talk about. Right, so what is the Data Party? In the process of generating reference genome assemblies, we make an assembly that is created and submitted to a public archive like an SDC. At this point, the assembly is deemed high quality and ready for public consumption, and that's where we enter. We take the assemblies and we compute a set of core genome analysis. So once all the assembly teams are done celebrating their genomes, here comes the after party. And we then share all the outputs in a public data repository. The goal is to complement the submissions to ENA to ENSDC, is to have a set of congruent, coherent analysis, and data types are derived from the assemblies. That can be shared to all research teams within our department, but of course, externally as well, because the goal is really about reusability and sustainability of the overall endeavor. And all of that is driven by the concept of a genome node publications. We, for every genome that we make, we publish a short publication, data note. And some of you may remember a presentation from Bethany Yates last year about biodiversity genomics and at PAG as well. So this short publication will tell about the assembly, species, assembly, the background, and we include some key statistics, key metadata about the species, the assembly, some assessment scores like BUSCO here, a summary of the assembly, and some graphical figures to help with the assessment as well, like this snake plot from the toolkit and a high-seq contact map. Another things that are needed to evaluate, compare assemblies on a similar level. And that's why we are hoping to expand that to many species. But really, there's a lot more we can do with genomes. A lot of different analyses that will produce a lot of beautiful plots. So the other party is also a set of analyses that many people would like to see on many genomes. That would include so there's typical GC content, but actually it's also of chemo analysis and repeat annotation. Read coverage is, for instance, very useful for taxonomic annotation and decontamination checks. Boost annotation is a very quick way to infer homologs across a bunch of species, and variant calls and sort of derived analysis like runs of homozygosity. So how are we doing that? We have chosen to use Nextflow as to implement the pipelines, and we follow the standards and ways of working of the NFCore community. And that means that all our pipelines are portable and cloud compatible. All software dependencies are packaged in containers. All parameters are in configuration files. We don't hard code any single specific paths or dependencies in the pipeline. And we can see already on GitHub external users using our pipeline and helping us improve them. All those pipelines are designed with reusability and shareability in mind. So for instance, following the NFCore best practice, all different software that we use in those pipelines are wrapped in individual modules that are shared back with the community. All the pipelines are available on a registry website that we built that reuses the NFCore infrastructure called our pipeline website. This will list all the pipelines that we have with their usage, documentation, different parameters, sample outputs, and so forth. All the code is on GitHub using the open source MIT license. And all pipelines can be cited through Zenodo DOIs. So at the moment, we've prioritized all the analyses that are relevant to produce the genome nodes. So that includes what we call the remapping pipeline that gives us alignments and coverage, plot toolkit, which gives us contamination analysis. And our pipeline that we call genome node is collates different assessment metrics about the assemblies. Those three pipelines are used in production. Have been now run on probably 1,500 species. We have a couple of pipelines being developed that are being rolled down in production gradually. So the one that looks at sequence composition and one that does variant calling and analysis. So where those data are. We have built a public data repository that we call GAP for Genome Third Party. So it's a public server hosted at Sanger. And if you go to the front page, you will see an overview of different species in assembly that we have, as well as a table that allows you to filter data. We are continuously running the pipeline on assemblies that come out of the Sanger Tree of Life assembly methods, but also some other assemblies from third parties. So the number that you see here are increasing every week. Then if you filter a species on assembly in a table, you will reach a page like this one. So for each assembly, you will have a reminder of the species name, the phylum, and the main table here lists all different pipelines analyses that we've run. So for instance, for this one, dolphin, we have the Blood Toolkit analysis available with a link to the Blood Toolkit viewer, got all genome statistics that are used to fit into the genome note, got the RIC alignments and the contact map. So I should add that it's not just a data repository. There's a set of data browser associated with it, just like Blood Toolkit, and we've got HiGlass3 for the contact maps, as well as other alignments. And here we've got variant calls. And the same page will also have the same sort of table that we see in the genome note. So really, this is a place where we can collate all information. The different links are links to data, so all the files can be downloaded. And that's going to serve doing large-scale analysis on all the genomes from the Earth Genome Project. So with that, I would like to thank my team, Informatics Infrastructure team within Tree of Life, but also the other assembly and faculty teams at the Sanger who had defining and driving this project. Really big thank you to the Nextflow community. I think it's a massive community with 10,000 people on the Nextflow Slack, and it's really the reason why we are able to develop pipelines that are such high quality and so scalable. And finally, all our partners and the umbrella projects, such as Digital and PGP. And thank you for listening.
So I've chosen quite a multi-part title as I want to talk about how we're building on the Genome Hub's code base used in Goat and Boat to begin to update our taxon-oriented sites such as MollusDB and LetBase via some of the standardized analyses run for Blob Toolkit and Genome Afterparty. Goat is a site that hopefully many of you are familiar with by now, where we hang various metadata for genomes on a tree. So very briefly, Goat is a searchable platform that lets us report on progress towards the goal of the Earth Biogenome Project to sequence all taxa, as well as other genome and assembly metrics. And we now have a YouTube channel as a place to share tutorials and other presentations. So if you want to know more about Goat specifically, you can head on over there. And we can search in Goat by taxon and rank to find values for the various attributes that we've indexed. And you can drill down to individual records to find out more. But coming back to the reporting side of Goat, as everything is on a tree, we can easily make a tree showing the current state of sequencing across all phyla. So here the green taxa have a chromosomal assembly, but the phyla in blue only have a scaffold level, purple contig level, and in grey there's no publicly available assembly yet. So the task of filling in gaps like these is shared across over 50 EBP-affiliated projects, including the Darwin Tree of Life, which has over 4,000 families on its wish list. But colouring these by sequencing status, we can see that much of the tree is green, as there are publicly available assemblies, and many other families are in progress or with samples collected. But at this resolution, it's hard to spot the details. So we can click to select a subset of rosids, for example, and get a more detailed view of the current status of individual families. Doing the same for Lepidoptera, we can see that for this part of the tree, most families either have a public assembly or in progress. So we can start to look in more detail at potentially the genera. And in fact, if we switch to the Psyche project, which is aiming for all European Lepidoptera, can see the progress of that project with many of the leaves that are not yet green in purple, as a lot of samples are already collected as the project progresses. But as fun as it is to kind of watch the colors change, as the sections of the tree become more complete, we've been thinking about how to explore what's inside the assemblies. And so last year I introduced Boat, which places busgoes and other assembly features on a tree. It uses the same code base as Goat, but with an emphasis on indexing assembly features rather than just taxon and assembly metadata. So here's an example fetching 100 KB windows for a single butterfly assembly. As with Goat, we can turn these into reports, for example, showing patterns of GC content along chromosomes, or patterns of read coverage, here highlighting the Z chromosome, which has half the coverage of the autosomes. So we can select to highlight this sequence to spot areas with higher coverage, and having identified the Z, we can switch back to see the pattern of GC compared with the background of the autosomes. So the data in BOAT have come from running the blob toolkit pipeline on many public assemblies. And these analyses include measures of coverage and GC content to help identify and isolate target versus contaminant and covariant data in preliminary genome assemblies. And there are BUSCO analyses to help identify regions of the assembly to use in sequence similarity searches to generate taxonomic annotations. So when there's a lot of contamination and short reads, these made for very colorful plots. But as the assembly quality is improved, running blob toolkit on finished assemblies should give much less exciting plots. But by breaking the chromosomes into windows, we can identify features such as the high mask content around the centromeric repeats in this Arabidopsis assembly. And the blob toolkit pipeline has evolved quite a bit over the last few years. So if you want to run it on your own data, I'd recommend checking out the Nextflow version at Sangatol blob toolkit as the latest and most effective way to run it. A problem we had with BOAT was that getting the blob toolkit analyses and some additional BUSCOs together ready for indexing, because although the data were only in a few locations, it made the import brittle enough that an update is now long overdue, so I'm really excited about having all the data in Genome Afterparty to make building resources like BOAT much more efficient. But as it wasn't fully up and running when planning the next steps after BOAT, we took a look at the primary data stored on the current version. And most of these can actually come from blob toolkit. So we started imagining a more efficient flow that started with blob toolkit, where we can feed the data into a gap-like S3 bucket. And in keeping with the theme of this section, I should mention that the logo I'm using is a placeholder and not the official logo. And then we can index the data and add an API and front end using the same Genome Hub's code base as GOAT and BOAT. Then eventually subsets of the data can flow out of GAP, be supplemented with clade level analyses and feed into taxon-oriented sites that also need to be updated, such as MollusDB and LetBase. But as there was a bit of a gap while Genome Afterparty got up and running, we sort of flipped things around by importing directly into a MollusDB S3 bucket, the idea that this could then be fed into GAP alongside analyses for all the other clades. So MollusDB itself is a site that we originally started a few years ago. Has a fairly small number of genomes plus some transcriptomes, but it's running on fairly inflexible tech. So we really wanted to bring it up to date and import a lot of the new genomes that have come along since. So step one was to move the archive data into an S3 bucket and then load newer data into S3 as well. So we could write a reusable set of scripts to index the data. And the result looks a lot like GOAT and BOAT, and includes the option to link the search results to relevant files so users can identify and access sets of raw data for whatever analyses they may want to run. But coming back to a more data-oriented table, one feature of mollusk assemblies is that they have low BUSCO scores. So here the numbers of complete and missing BUSCOs show many assemblies have over 1,000 missing loci. We can view the BUSCO completeness values as bars on a tree and see that across the tree, many of the lineages have low scores, and the ones with the higher scores correspond roughly to those that are included in the ODB10 set. But putting this on a scatter plot of mollusk versus metazoa BUSCO completeness, a few bivalves are on the line of expected mollusca BUSCO score right at the top right, but many have considerably lower completeness than expected based on the metazoa value. We can drill down into the data at a species level, adding a column with the actual BUSCO loci. And because the values have filled up the tree, just like in GOAT, when we look at the class level, we can see that there are large numbers of BUSCO IDs that are not found in any assemblies in most classes. And even in bivalvia, which have the most complete assemblies, there are 4 loci that aren't in any assembly. And we can click through to find out which they are. So MollusDB provides a platform to assess expectations for the completeness of new assemblies and potentially derive more appropriate subsets for use in assessing completeness. But rather than dwell on the challenges of using BUSCOs in mollusks, we can put the ones that are found to use as a set of conserved loci. So here, painting the chromosomes of a razor clam Cyanobacula constrictor with ancestral linkage groups identified by Marcela Juliana Silva using SynGraph. So highlighting individual series here helps to show chromosomal rearrangements relative to the ancestral state. So for example, here, ALG1, it's found on 2 separate chromosomes, but it's also appears to have fused with ALG16, where it is found as large blocks of loci from both these ALGs are present in the longest and 10th longest chromosomes. So we can check the position of this species in the tree and can find the closest sister taxon that we have data for is Azorhinus camosolon. And we can use the fact that we can match busco IDs across assemblies to generate an Oxford plot. So here we can see that while there's been a lot of rearrangement within chromosomes, most busco loci have stayed in the same linkage group since the common ancestor of this species pair. So highlighting ALG1 again, you can see that it's present in a single chromosome in the closest taxon, which is shown on the y-axis here. And the same is true for ALG16. So rearrangement of these in Cynonobacula appear to have been recent. So to get an alternative view of the same data, we can switch to a ribbon plot, which helps to emphasize loci that have changed location between these 2 species. So again, we can highlight the individual ALGs, highlighting that the separate groups in Azorhinus, shown at the bottom of the plot, shown at the top of the plot, have shifted into 2 mixed chromosomes in Cynonobacula. And selecting only those loci that are found on specific chromosomes, we can reduce the noise in the plot and more easily follow the paths of the specific loci. So searching for only loci that are connected to the longest and 10th longest chromosomes shows things more clearly. And so it seems likely that there was a fusion of ALGs 1 and 16, followed by some rearrangement before a subsequent fission gave the chromosomes that we see in Cynonobacula now. So in this view, we can easily focus on individual loci to discover the busco genes involved, and then click through to find more information about the corresponding features in each assembly. But having followed that example through, I realized that it doesn't have all the information I wanted to show on the next screen. So I'll jump to a feature in a different species to show you what you may see when you click through one of the table rows. Here, the record pages are similar to GOAT with taxon and assembly links. And as it's a feature, there's a diagram of its position in the chromosome and where available links to entries in ensemble sites, which take you out of MollusDB and into the relevant ensemble genome browser. And in exchange from the previous MollusDB, which had a local browser for each assembly, this new version is focused on assemblies that are publicly available elsewhere, so we can make full use of available external resources. Now, this summer, we were fortunate to have 2 Google Summer of Code contributors working on projects related to genome hubs. Deep Nyak's been working to bring natural language search to GOAT to make it easier to formulate queries and discover data. But I don't have time to go into this in detail, but for the other project, Rohan Basagod has been working on integrating Kinfin into genome hubs, and this is a feature that we're keen to add to MollusDB. As it's updated and extended the Kinfin code base, originally written by Dom Lech, Kinfin can be used to process clustering from AutoFinder and relate the clustering data to user-defined sets to provide insight into loci that are associated with particular traits. When run conventionally, the user needs to provide an AutoFinder clustering, some divisions into sets of taxa and to compare, and then Kinfin will generate a set of tables and plots slicing that data up in various ways. And to make this usable in the context of MollusDB, we've devised a workflow where the clustering is run once and held on a MollusDB server, and then the subsequent steps can be run via an API with the user specifying how the data should be grouped and requesting specific outputs from the full set generated by the analysis. To make this work from the UI, we've added the ability to bin any tabular data results by adjusting the parameter that controls how many bins to use in a similar way to the histogram plots and category coloring in existing reports. Now, to output, this outputs a table of binned results, which can be passed to the Kinfin API. And the next steps are a work in progress, but soon Kinfin will be available as another report type for any MollusDB search results. So looking beyond mollusks, we set this up to be reusable across any clade. So we're looking to rebuild LetBase along similar lines and already have a request for IsopodDB and possibly others to follow. LetBase was running on similar code to the old MollusDB, but it's had to be retired as the server had some technical issues. At the last count, had annotated assemblies for 27 species, which is a long way short of the current status, let alone the ambitions of Project Psyche. So as for MollusDB, we've set up an S3 bucket and now need to make sure that all the standardized analyses are collated either here or on the main S3 bucket, so the main GAP S3 bucket, so we can run the scripts used for MollusDB and then bring the next version of LetBase online. And so with that, I'd just like to say thank you to everyone who's contributed to Goat, Boat, Blob Toolkit, GAP, MollusDB, and LetBase, and in particular to the people that have contributed to the particular code, the data, and the analysis that I've presented here. So thank you.
Good afternoon, everyone, or good morning, good evening, depending where you are. So this is a session about genome analysis tools and resources. So we had 3 talks, and joining with me today are Marco Suito, so he's a postdoc at the University of Firenze in Italy, in the group of Professor Claudio Chioffi. During his PhD, he worked on genomics of marine organisms, and as you've seen, now he's moved on the collaboration with the Giulio Formenti at the Vertebrate Genome Lab at Rockefeller, and working on Galaxy pipelines. Richard Chellis is a senior mathematician working at Wellcome Trust Constantine Institute in Mark Baxter's team. He was previously at the University of Edinburgh. He's lead developer of projects such as Blood Toolkit, Genome Hubs, Goat, and you've seen MollusDB and Boat and many applications in the Genome Hubs universe. So we have a few questions on the Slido already. We've got one for you, Marco, from Marcela Uliano da Silva. So, is there a genome size limitation for the use of Galaxy annotation pipeline?
Hi everyone, and yeah, basically I test the workflow even for raw deers, so they have like a genome size around 3 gigabytes, and it was working, but before we should need some implementation in the config file in order to give like some additional parameters to star, because the stopping point in star that is requiring an higher RAM usage, and so basically it's working, but not as a default. But we will work on to make it this as default, at least for genomes around 3 gigabytes. That's like the one of the mammals, and then of course for the other genomes with higher genome size, it will be more complicated and requiring higher RAM usage.
And next one from Dorothée Huchon, from Marco again. Do you compare your pipeline with Breaker 3?
Yes, I have compared the pipeline with Breaker 3, and basically looking at the Jacquard index, so the index of similarity among 2 datasets, and using as a reference the one that has been provided by the NCBI RefSec notation pipeline. We have a higher level of Jacquard index with the EGAPix rather than the other rather than Breaker 3. I mean, and both it was I checked the result, and it was like this with higher value even at the gene and taxon level. So yes, it's working better, and I mean, yeah, should be tested, of course, more and more, and we should wait for feedbacks from you even.
Moving on to Richie's talk, we got one question from Kerstin Rao. So genome browsers usually have the concept of federated data, trackers, these sort of things. So will it be possible to have federated data on Boat and Goat?
Yes, I guess there's kind of 2 angles to it. Certainly, I can see it'd be very useful to have the data files for the track hubs. They can live on either on MollusDB, or I guess more broadly, they could be part of GAP for any species. So they could be pulled into another browser. I guess there's also the level where the the Genome Hub sites could potentially make those data that are in a Track Hub searchable. And so it would be possible to work it the other way around and have, so for example, if it's depth of RNA-seq coverage, for example, along a chromosome, then could index those values to find the regions of higher RNA-seq coverage, or even potentially to find the locations where the coverage shifts from high to low in order to kind of identify regions corresponding to gene boundaries. So there are a few ways to do it, but I think certainly at the simplest level to incorporate, I think they would be very useful data to include, actually have the Track Hub files available for some of the common analyses to be pulled into other browsers.
Question from Tom Brown to Rich, but maybe related to me as well. So about BUSCO coverage, so whether you're putting those values from the after-party space or computing those in parallel.
Yeah, so as far as possible, yes, putting them in from genome after-party space. There's been a few texts where we've kind of been trying to get ahead of the genome after-party pipeline and prioritize specific assemblies. But yeah, even those data we're hoping to then push back into genome after-party. Just so yeah, we're trying to avoid duplication of compute there.
I can probably answer that and some of Marcella's question at the same time, who's asking about coming on next step for the GAP and both. So currently in production within the genome as a party, we've got all the principal assemblies coming from the Sanger, and the other ones we're doing genome nodes for. We've got past data for a bunch of other assemblies, especially coming through the Genome Hub toolkit server. And we're now trying to roll out this system and adding more assemblies. So depending on the kind of type assembly, and I may answer Tom Brown's question about whether we do every assembly or not. So the primary assembly, we have remapping and completeness statistics and quality statistics such as Kmer and BUSCOs. May have variant coding as well, whereas an alternate assembly will only have the completeness statistics, but not variant coding done. So we, in this process of ruling things out, making sure always we've reached that we only computing things once and having the data, then the outputs displayed on multiple sites, available for download and analysis. So Tom Brown is asking how often will all the statistics and plots need to be recalculated as new tools databases are updated. I think that's, especially for BUSCO, that's an open question, because with the latest version of BUSCOs that keep coming every few months, switch to miniprot as the backend. Maybe a switch to a new ODB at some point. I don't think we have an answer for that yet, Rich, do we?
Yeah, I think in this context, it's kind of important that we manage to keep things consistent across all the analyses that we have. So I guess it will be important to update at times, but probably not to chase all the incremental updates and risk having different versions across different things that are available. So I think it might be something that happens after a couple of years, rather than every new development requiring a new analysis. So yeah, we're trying to put out a set of resources where people don't have to repeat analyses, but yeah, I think there will inevitably be times when people want to run a different version of a tool for a specific reason. And I think if we tried to catch every assembly through every version as these things are improved, then we will end up running more compute rather than less.
We actually extend the question to Marco. So your pipeline is running certain tools, certain versions. How do you foresee and for given Cladle project, assemblies will be released at different time. And are you planning to upgrade the version of software using your pipeline? But so how do you foresee having consistent annotation, consistent data across say all Clade, all the set that's being produced, over a certain period of time?
Yeah, at the moment we are in the we have released the alpha version. So basically, there are still some points that should be implemented. Of course, as I told during the presentation relating to the non-coding part. So when we conclude even introducing the next release, basically we will just annotate basically. In order to improve we will start with the VGP genome assemblies that have been already assembled, and then we will go further on that. And that's basically we will cover, of course, all the orders of the vertebrates.
Tom is asking how long does Rich, how long does it take for the completion of each assembly? And whether we'd like to have multiple version of multiple version of the same species within both.
Yeah, so yeah, I guess yeah, the computation time is yeah, very much dependent on the assembly size. So it's, I mean, most of the assemblies in MollusDB are running in kind of less than a day, I'd say. But yeah, for some of the larger assemblies, they are taking quite a considerable amount of time. And I think that there are some open questions within certainly within sites like MollusDB, where potentially for boat, I think the goal will be to just have a representative assembly for each species, whereas for something like MollusDB, it becomes more about questions that the community working on that group of taxa want to ask. And so I think that's where comparison of different assemblies within the same species becomes perhaps more within scope of the tool. So I think it's more likely that new versions will be included there. But I think more generally, kind of the priority order for running assemblies through GAP, as been talking with Matthew, is kind of starting with the kind of, I guess, what is currently gold standard EBP quality assemblies. So it's unlikely that we should have to update those particularly regularly, as they're already chromosomal and near complete assemblies. So I think it's once we start including a more wider set of assemblies, we're potentially including more scaffold level assemblies that we'll have to look at replacing or updating to new versions as they become available.
Going on that, I have a question about related to pangenomes. So say we've got multiple assemblies of individuals or multiple populations of same species or very closely related species. Could you easily imagine lifting over some knowledge annotations from one assembly onto the others, and have each assembly improve the other annotation at the same time? Marco, is that something that you've come across or found interest in several projects?
Yeah, it's something that we think about, and that, of course, should be taken in consideration, the presence of pangenome and the presence of sense variation along all the genomes. And yeah, of course, this will be some future points even after the release of the complete pipeline, and how to recognize this at the pangenome level, to implement this, of course.
And reach the synteny view, for instance, would look very different if you're comparing very closely related, species assemblies versus different species. So how do you imagine that framework working?
Yeah, I guess it's as with all the tools, it's the principle within I guess the principle carried forward from Goat is just to make everything available for any comparison. And I suppose it is incumbent on the user to work out when those tools become less informative. And so I think I don't see an easy way of limiting that, although it's there's potentially interesting information to be had. So, for example, taking the set of Busco loci across all mollusks, that breaks down very quickly, particularly in things like cephalopods, where just the very fact that that's breaking down so quickly makes the plot informative in a way, even though they're then not necessarily looking to dive deeper into the information that's presented. So I guess it's just about taking things at different levels.
There's a question from Eric Tvet. I'm not sure exactly who's asking the question too, but it's about whether we have any plans for automated contaminant removal from the toolkit results in our next flow pipelines, or does it always require manual review? I think this probably refers to the Sanger flow toolkit team. So the genome assembly teams, I think, always do a manual review at this stage. That's the way they work to ensure that we generate the best quality possible assembly at the end. So I think you would have to ask them how they would imagine automated contaminant removal from pipeline. Maybe refer you to the Biodiversity Genomics Academy session that was held a few weeks ago. So there are tutorials and videos about how they're doing creation in the team. All right. We're almost at the end of the session. Does anyone in the chat have a last question to ask? Otherwise, Marco and Rich, actually, I could have asked you, do you have questions as well?
Not at the moment, and just thank you.
All right. So with that, I will thank all the speakers. And as Mark said, a thunderous applause for everyone. Thank you everyone for listening, and see you in a couple of minutes for the next session.